Hi class, my name is Kai.
And I'm Jean, and today we're going to be identifying an unknown bacteria.
Why is it important to be able to identify bacteria? Being able to identify the bacteria is crucial in medical diagnostic, food preservation, and pharmaceutical uses. For instance, being able to identify the bacteria gives doctors and microbiologists the ability to find appropriate antibiotic to prescribe to patients with bacterial infection. In pharmaceutical industry, the identity of bacteria could provide useful information to create vaccination against that pathogen. For food industry, researchers could find microbial contaminant that is responsible for food spoilage. The purpose of this lab is to isolate and identify an unknown bacteria given by the instructor. The student will perform inoculation, incubation, isolation, examination, and identification to identify the unknown. Can you go to next slide, please? For materials and method, first we're going to serial dilute the sample to obtain a viable titer. With that viable titer, we're going to pick one colony and perform various isolated strips to obtain a pair extended cultures. In addition, gram stain is going to be performed on that isolated strip to determine the cellular morphology of the unknown. One has been confirmed through colonial and cellular morphology, that is a pair extended cultures. The sample is going to be placed through several biochemical tests and selective differential media. The result from the biochemical and selective differential media is going to be placed in an API 20E test and Kirby Bauer assay. Using those information, we're going to refer to the Bauer manual in order to identify the bacteria and other identification result from the API 20 test. Could you go to the next slide, please?
And for the results in week 4 and 5, we performed isolation streaks and we also performed a gram stain on that isolation streak. In order to determine the colony and cellular morphology, we looked at the isolation streak first for the colony morphology, and there was almost no single colony present on the isolation streak, but there was that small speck that you see right on the screen. And that gave us a good idea of the colony morphology. It was very irregular shape. It was a little bit raised, kind of shiny, almost looks like slimy, rather opaque, but transparent in some places. And then, as you can see in the gram stain, it is gram negative, and the all of the you can see the uniformity, the purity of the cells present. They are bacillus shape, but they're also kind of circular, so they're like cocoa bacillus. um they're in a staphylococcus arrangement or staphylo arrangement. Some are diplo, some are singular, but they're basically all clustered together. And as we can see here, they're rather pure cultures just because they are rather uniform throughout. In week 6 and 7, we performed a biochemical test on these on this isolation streak that we just saw, and essentially all biochemical tests and selective media came out positive except for gelatin hydrolysis and oxidase. So our bacteria contains the enzymes catalase, urease, amylase, nitrate reductase. It's able to ferment complex sugars such as lactose, sucrose, and glucose. The OF medium test tells us that it's a facultative anaerobe. The EMB McConkey agar gives light to the fact that it rapidly metabolizes lactose, as we can see from the EMB agar, the green sheen, and the McConkey agar gives a red halo around the bacterial growth. The blood agar shows us that it performs gamma hemolysis. So it won't lyse the cells, it will just grow on the blood. And the mannitol salt agar test, this is the media that gave us an unexpected result, because usually um, our expectation based on these biochemical tests was E. coli, and E. coli is usually not halophilic. Only halophilic bacteria may grow on mannitol salt agar, and also some that ferment mannitol. So we also know that um, our bacteria can metabolize mannitol, and that it's halophilic. And as a result of this expectation of it being E. coli, some media for isolation would be the sorbitol, McConkey agar, and lauryl tryptose um, broth. Both of these are would be perfect for our bacteria, assuming it's E. coli, since these promote the growth. They have compounds in there that promote the growth of of E. coli, like lactose, and inhibit the growth of other bacteria like gram-positive bacteria or anything that isn't E. coli. In week 8 and 9, we performed an API 20E test and we looked at the Burgess manual to identify the bacteria. This was rather easy because we just had to fill up the wells and get the results. The results for the API 20E test were that it matched 97.4% with E. coli, and the Burgess manual also matched it with E. coli, as you can see from the 5 steps that were given. So then the API 20E test and Burgess manual matched up. In week 10, we did the antibiotic resistance with the Kirby-Bauer assay. And as you can see on the image right there, it was resistant, our bacteria was resistant to 3 antibiotics, being bacitracin, erythromycin, and penicillin, and susceptible to the other 3. And now for the problematic discussion. We based off our results on this study done identifying E. coli. The citations that I have there refer to a study done in 2006 that performed identification tests on E. coli. So essentially, an EMB agar, these test results, yes, you can see on the agar plates and the table, an EMB agar that has a green metallic sheen is positive for, or gives good indication rather for E. coli, and any lactose fermenting intense pink colonies on the McConkey agar, any red halo on the McConkey agar is also a very good indication of E. coli, as said on the bottom right. And right next to it on the table, as we can see, E. coli should have a positive indel test, a positive methyl red test, a negative Volksperskauer test, and a negative citrate test, which all matched our results. We had the metallic sheen, we had the red halo, we had a positive indel, positive methyl, and negative citrate, negative Volksperskauer. So it basically perfectly matched with the bacteria, except um, the mannitol salt agar test being positive. However, there are studies being done of a specific strain of E. coli being halophilic. E. coli strain B has been known to survive in high salt concentrations. So there's a very big probability that our bacteria is this E. coli strain B. And for further methods of isolation and identification, we could use the PCR chain reaction, which is further used in genetic sequencing to really get a good idea of what our bacteria is. And also the spot indel test, which if a positive is given, is a very, very, very good indication that whatever we have is E. coli.
For the environment of E. coli, so E. coli is usually found in intestine of warm-blooded animals, contaminated water and food, and basic any food products that has been contaminated with fecal matters. Could you go to the next slide, please? The importance of E. coli. E. coli plays a crucial role in maintaining a smooth digestive circulation. It prevents bacteria in large intestine from growing uncontrollably. In addition, it also provides protection against harmful pathogens in the digestive tract. E. coli has the ability to divide rapidly and is frequently used for a model organism. Factors such as the ability to grow fast using cheap media is crucial to perform genetic manipulation and are favorable in molecular genetics. For relevance of the E. coli plays a crucial role in modern biotechnology as well. It often used to store DNA sequences from other organisms. These bacteria will use to produce and test proteins. Lastly, E. coli break down food and produce vitamin K. However, some E. coli strain could have devastating effect on the body and cause diseases such as hemolytic uremic syndrome, which can lead to kidney failure and death. Could you go to the next slide, please?
And here are all of our wonderful references that have given us information to complete this presentation. And thank you for your attention. Are there any questions? Please ask questions. This is for your grade. Thank you.